Welcome back, everyone, for another deep dive. This time, we're really going where few have gone before, exploring the fascinating world of pharmacogenomics. That's right, folks. Get ready to unlock the secrets of how your unique genes can actually impact how you respond to medications.
It's personalized medicine, but at the DNA
All
level.
right, so to help us decode all of this, we have a couple of really interesting sources lined up. First up, we have a YouTube video that dives deep into the world of SAMP signaling.
And we'll also be taking a close look at a research paper focusing on those fascinating thyroid hormone receptors.
Our mission today, to uncover how variations in your genes, especially those coding for receptors, can significantly impact how medications actually work for you.
It's like imagine a world where treatments are tailored to your specific genetic makeup.
Okay, so before we get too far ahead of ourselves, let's break this down a bit. For those who might be new to this whole concept,
Mhm.
could you give us a quick rundown of what pharmacogenomics is all about?
Well, it all boils down to this. Our genes hold the blueprints for how our bodies function, including how we process medications. And just like we all have unique physical traits, we also have variations in our genes that can affect how our bodies respond to drugs.
So basically, our DNA can influence whether medication is effective for us, or if we might experience side effects.
Exactly. It's like a puzzle where each person has a slightly different set of pieces.
That's a great way to put it.
Hmm.
So where do these receptors fit into all of this?
Receptors are these key proteins found on the surface of our cells. They act like gatekeepers controlling what goes in and out. And many medications work by binding to specific receptors to trigger a desired effect.
So if I'm picturing this correctly, these receptors are kind of like cellular doorways, and medications are the keys trying to get in.
That's a fantastic analogy. And just like keys, some medications might fit perfectly, while others might not work as well, or could even cause unexpected side effects due to variations in these receptors.
Okay, that makes sense. Now, the YouTube video we have uses a really interesting example to illustrate this point, something about heart muscle cells.
Ah, yes. The cardiomyocytes, they have these two different receptors, the beta-1 adrenergic receptor and the EP4 receptor. Both respond to different signals. But here's the catch. They both trigger the same cascade involving CAMP, a crucial messenger molecule inside the cell.
Okay, so same messenger, but different outcomes depending on which receptor gets activated. Sense and plot twist here.
You got it. When the beta-1 adrenergic receptor is stimulated, it increases the force of heart contractions, but stimulating the EP4 receptor doesn't have the same effect, even though it activates the same SAMP pathway.
Wait, that seems counterintuitive. Why would activating the same pathway through different receptors lead to such different outcomes?
That's where the concept of microdomains comes in.
Microdomains. All right, I'm all ears.
Think of them as tiny compartments within the cell where signaling molecules like CAMP can be concentrated, leading to localized effect.
So it's not just about the messenger, but also about where the message is delivered within the cell.
Precisely. It's like having different mailboxes for different departments within a company.
So even though both receptors activate CANMP, they might be doing so in different areas of the cell. And that explains the different effects on heart contractions.
Exactly. And that's where pharmacogenomics comes into play.
Okay, how so?
Well, imagine this. If you have a genetic variation that alters the structure of one of these receptors, it could affect the formation or function of these microdomains, potentially altering the drug's effect.
So my version of the beta-1 adrenergic receptor could be slightly different from yours. And that could mean our heart muscle cells react differently to the same medication.
Precisely. And that's a key concept in pharmacogenomics. These subtle genetic differences can have a significant impact on how our bodies respond to drugs.
This is blowing my mind. So we've got these microdomains directing traffic within the cell. Are there any other key players in this intricate signaling dance?
Absolutely. The YouTube video also mentions A-kinase anchoring proteins, or ACAMPs.
AKQs.
They act as scaffolding proteins, ensuring that another crucial enzyme in the CAMP pathway, protein kinase A, or PKA, is in the right place at the right time.
So AKPs are like the organizers of the cell, making sure everything runs smoothly.
Exactly. And to make things even more complex, there are different types of PKA, each with varying affinities for specific AKPs.
Wow, it sounds like even tiny variations in these AKPs could throw off the entire signaling cascade.
You hit the nail on the head. And this is where pharmacogenomics comes full circle.
How so?
Imagine if a genetic variation affects the function or localization of a specific AKP. This could disrupt the entire CAMP pathway, leading to an altered drug response.
Okay, this is all starting to make sense now. So we've explored these microdomains and AKPs, these tiny cellular components that play a huge role in drug response. Now I'm curious to see how these concepts apply in a real-world scenario.
Perfect timing. Let's shift gears and dive into the world of the thyroid hormone receptor, using those insights from the research paper you mentioned.
Okay, I'm ready to explore the world of thyroid hormones. What makes this receptor so fascinating from a pharmacogenomic perspective?
Well, the thyroid hormone receptor, or TR, plays a crucial role in various bodily functions, like brain development and metabolism. What's really interesting is that two genes, TREA and 2 rate code, for multiple TR isoforms.
Hold on, remind me again what exactly are isoforms?
Think of them as slightly different outfits for the same protein. They originate from the same gene but are assembled in slightly different ways, leading to subtle variations in their functions.
Okay, so we have multiple TR isoforms, each potentially with a slightly different role. Why does this matter for pharmacogenomics?
Because the research paper highlights that these TR isoforms have distinct expression patterns across different tissues. For instance, you might have more of one isoform in your liver and another dominant in your brain. This means the same thyroid hormone can exert different effects, depending on which TR isoform is present in a particular tissue.
That's so cool. It's like having specialized receptors for different parts of the body.
Exactly. And to add another layer of complexity, the research paper delves into the hormone binding domain, or HBD, of the TR.
HBD.
Mutations in this domain can lead to resistance to thyroid hormone, or RTH.
RTH data. That sounds serious. What happens in this condition?
Individuals with RTH have a decreased sensitivity to thyroid hormone, even if their hormone levels are normal or even elevated.
So it's like their body is sending out the signal, but the cells aren't receiving it properly.
Yeah.
Because of those mutations in the receptor.
Exactly. And this ties back to pharmacogenomics.
Okay, I see the connection.
If you have a variation in the gene coding for your TR, this variation could alter the receptor's structure and function, impacting your sensitivity to thyroid hormone and potentially affecting your response to thyroid hormone replacement medications.
So if I have a variation in my TR gene, I might need a different dosage or a different medication altogether, compared to someone with a different variation.
Exactly. And that's precisely why understanding these individual genetic variations is crucial for personalized medicine. It allows us to tailor treatments to each person's unique genetic makeup.
Incredible. It seems like we're just scratching the surface of how complex and individualized our responses to medications can be. Are there any other surprises in store when it comes to these thyroid hormone receptors?
There are. The research paper also touches upon something called non-genomic effects of TRs.
Non-genomic.
Uh-huh.
That sounds intriguing.
Traditionally, we think of receptors like TRs as working within the cell's nucleus directly affecting gene expression. However, non-genomic effects suggest that these receptors might have actions outside of the nucleus as well.
So even more complexity. It seems like we still have much to learn about how these receptors work.
Absolutely. It's a dynamic area of research with many exciting discoveries yet to be made. But to really understand the implications of these genetic variations, we need to zoom in even further and explore how TRs interact with our DNA.
Okay, I'm ready to dive deeper into the molecular world. What can you tell me about how TRs regulate gene expression?
This is where it gets really fascinating. TRs can actually function as both gene silencers and gene activators.
Right, they can do both.
Yep. It all depends on whether the thyroid hormone is bound to the receptor.
How does that even work?
So in the absence of thyroid hormone, the TR binds to DNA and represses or silences gene expression. But when the thyroid hormone is present, it binds to the TR, causing a shape change in the receptor. This allows it to activate gene expression.
Oh, it's like a sophisticated on-off switch controlled by the presence or absence of the hormone.
Exactly. And this process is further fine-tuned by other molecules called cofactors, specifically core pressors and coactivators.
Core pressors and coactivators.
Yeah.
So what do they do?
Think of them as partners in a cellular dance, helping the TR orchestrate gene regulation.
Okay, so they help control the flow.
Yes. When thyroid hormone binds to the TR, it prompts core pressors to detach and coactivators to attach. This shift in partners further modifies the TR's activity, either enhancing or repressing gene expression.
Wow, it's like a carefully choreographed molecular ballet with the TR leading the way.
I like that analogy. And to make things even more intricate, the research paper mentions specific DNA sequences called negative thyroid hormone response elements, or NTREs.
Can do.
These sequences can further modulate the TR's activity, adding another layer of complexity to this intricate dance.
NTREs. So even the DNA itself is part of this intricate regulatory system. It's mind-boggling to think about all these different components working together.
It truly is, and this complexity highlights a crucial point. Even small variations in the genes for TRs, these NTREs, or the cofactors involved, can have a ripple effect throughout the entire system. This brings us back to the core of pharmacogenomics.
right? Even a tiny change in the DNA can affect how our bodies respond to medication. This is all incredibly fascinating. But I'm curious to know what the consequences of these variations in TRs are in the bigger picture.
That's a great question. The research paper emphasizes the critical role of TRs in development, especially in brain development, hearing, bone growth, and metabolism.
So it's not just about how we respond to medications as adults. It's about how these variations can influence our development from the very beginning.
Exactly. And to illustrate just how impactful these mutations can be, the research paper mentions the VRBA oncogene. It's a mutant form of TI that can actually contribute to cancer development.
Wow, a mutated TR can contribute to cancer. That's a stark reminder of how crucial these receptors are for our overall health.
It is, and it underscores the incredible potential of pharmacogenomics. Imagine being able to identify these mutations early on and develop targeted therapies that address the root cause of the problem.
That would be revolutionary. This deep dive into the microscopic world of TRs and gene regulation has been truly eye-opening. Now I'm eager to connect all of this back to the real-world applications of pharmacogenomics.
I think you're ready to see how this knowledge is already being used to personalize treatments and improve patient care. Yeah, it's really mind-blowing to think that we're moving away from this one-size-fits-all approach to medicine and stepping into the era of truly personalized treatments.
It's about time. But I'm curious how this knowledge is being applied in practice. Can you give me some concrete examples of how pharmacogenomics is making a difference in patient care today?
Absolutely. One area where pharmacogenomics is already having a major impact is in cardiovascular disease. Remember those beta-adrenergic receptors we talked about earlier, the ones in heart muscle cells?
Yes, the ones that play a role in regulating heart contractions.
Exactly. Well, there's a lot of genetic variation in the genes that code for these receptors, and these variations can significantly affect how people respond to beta blocker medications, which are commonly prescribed for high blood pressure and heart problems.
So are you saying that some people might not respond as well to beta blockers because of their genes?
That's right. For some people with certain genetic variations, the standard dose of beta blockers might not be effective, while others might experience more side effects at the same dose.
So by understanding a patient's genetic profile, doctors could potentially personalize the dosage, or even choose a different medication that would work better for them.
Precisely. It's all about maximizing the benefits of the medication while minimizing the risks. Pharmacogenomics gives us the tools to fine-tune treatment strategies based on individual genetic variations.
That's truly personalized medicine in action. What other areas of medicine are being revolutionized by pharmacogenomics?
Another great example is in the field of cancer treatment. Some chemotherapy drugs are metabolized by an enzyme called CYP2D6. And there's a huge amount of genetic variability in how people produce and process this enzyme.
So some people might break down the drug very quickly, while others break it down slowly.
Exactly.
That sounds like it could lead to significant differences in how effective the treatment is.
And it's not just about effectiveness, it's also about toxicity. If someone metabolizes the drug very slowly, they might be at a higher risk for side effects because the drug stays in their system longer. On the other hand, someone who metabolizes it quickly might not get enough of the drug to be effective.
So how do we address this issue?
That's where pharmacogenomic testing comes in. By analyzing a patient's CYP2D6 gene, doctors can gain insights into how they're likely to metabolize the drug and adjust the dosage accordingly.
That's incredible. So pharmacogenomics is being used in cardiovascular disease cancer treatment. Are there other areas where it's making a difference?
Absolutely. Pharmacogenomics is also being used to guide treatment decisions for a wide range of conditions, including mental health conditions, infectious diseases, and even pain management. The applications are expanding rapidly as we learn more about how our genes influence our responses to medications.
This is truly revolutionizing healthcare. But with all this talk about personalized medicine and genetic testing, I have to ask, are there any challenges or ethical considerations we need to address?
That's a crucial question. You're right, there are challenges that need to be considered as this field continues to grow. One of the biggest challenges is access to pharmacogenomic testing. It's not yet readily available to everyone, and it's not always covered by insurance plans. This means that there are disparities in who can benefit from this technology.
And I imagine that not all healthcare providers are equipped to interpret the results of these tests and integrate them into their clinical practice.
That's another key hurdle. There's a definite need for more education and training for healthcare professionals on the principles and applications of pharmacogenomics. We need to ensure that clinicians understand how to use this information to make informed treatment decisions.
It sounds like we're opening a door to a whole new world of possibilities. but we need to proceed with caution and responsibility. What does the future hold for pharmacogenomics?
I think we're on the cusp of a truly personalized medicine revolution. With advancements in technology, genetic testing is becoming faster, cheaper, and more accessible.
So you're saying that in the future, potentially everyone could have their genome sequenced and use that information to guide all their healthcare decisions.
That's the vision. Imagine a world where medications are carefully chosen and doses are fine-tuned based on your unique genetic makeup. It's a future where medicine is truly personalized and tailored to each individual's needs.
That raises some interesting questions, though. What happens if we discover a genetic variation that makes someone resistant to all currently available treatments for a particular condition?
That's a valid concern. And a reminder that pharmacogenomics is not a magic bullet. It highlights the critical need for ongoing research and drug development. We need to continue expanding our arsenal of therapeutic options to address the incredible diversity of human genetic variation.
Right. Even with all the amazing progress we've made, we're still only scratching the surface of understanding the complexities of the human genome and its impact on how we respond to drugs.
Absolutely. It's a continuous journey of discovery, and there's so much more to explore. But I think this deep dive has given us a good foundation for understanding the power and potential of pharmacogenomics.
It definitely has. We've covered a lot of ground, from the intricate world of microdomains and AKPs to the fascinating complexity of the thyroid hormone receptor, and we've explored how this knowledge is already being applied to personalized treatment strategies and improve patient care. It's clear that pharmacogenomics is revolutionizing the way we approach healthcare.
I couldn't agree more. It's a paradigm shift that's empowering both patients and healthcare providers, with more precise and personalized tools for achieving better health outcomes.
So as we start to wrap up our deep dive, what stands out to you as the most significant takeaway from all of this?
I think what strikes me the most is how this knowledge is changing our perspective on healthcare as a whole. We're moving away from a reactive approach where we wait for symptoms to appear before intervening, towards a more proactive and preventative approach.
It's like we're finally recognizing that each individual is unique, and their genes play a vital role in their health and how they respond to medications. It feels like we're cracking the code to truly personalize medicine.
It's an incredibly exciting time to be in this field, and as we continue to unravel the secrets of our genes and how they interact with medications, we have the potential to make healthcare more effective, safer, and more personalized than ever before.
Wow, this has been an incredible journey through the world of pharmacogenomics. It's clear that this field is revolutionizing healthcare. But with all this talk about the future, I'm wondering what steps individuals can take right now to potentially benefit from these advancements.
That's a great question. It's important to remember that pharmacogenomics is still a relatively new field, and its integration into standard medical practice is ongoing. But there are definitely things you can do to be proactive.
So what's the first step? Should everyone rush out and get their genome sequenced?
Not necessarily. The first and most important step is to have an open conversation with your healthcare provider.
Good old-fashioned doctor-patient communication.
Exactly. Talk to your doctor about pharmacogenomic testing and whether it might be relevant to your individual health conditions or the medications you're currently taking.
So it's not a one-size-fits-all recommendation. It's about having a personalized discussion with your doctor to see if this type of testing aligns with your needs and health goals.
Precisely. Pharmacogenomics is all about personalized medicine, and that personalization begins with a conversation between you and your healthcare provider.
And if someone does decide to undergo pharmacogenomic testing, what's the next step?
Well, if you do undergo testing, it's crucial to have a thorough discussion with your healthcare provider about the results. Make sure you understand what the findings mean for you and how they might influence your treatment plan.
Right, because a genetic test is just one piece of the puzzle. It's important to consider all the other factors that contribute to your overall health and well-being when making treatment decisions.
Absolutely. It's a collaborative process between you and your healthcare provider. Pharmacogenomics provides valuable insights, but it's not meant to replace the expertise and judgment of your doctor.
So now let's circle back to the topic of those thyroid hormone receptors. The research paper we discussed earlier highlighted some interesting insights into their role in disease and development. What were some of the key takeaways there?
One of the most striking points was the discussion of resistance to thyroid hormone, or RTH. As we mentioned earlier, it's a condition where individuals have mutations in the thyroid gene, making them less sensitive to thyroid hormone. And what's fascinating is that the severity of RTH can vary greatly from person to person, depending on the specific mutation they have.
So even within a single condition, there's a whole spectrum of genetic variation that influences how the disease manifests itself.
Exactly. It underscores the importance of personalized medicine. By understanding these individual variations, we can tailor treatments more effectively, taking into account each person's unique genetic makeup.
It's like the difference between having a custom-made suit that fits you perfectly
Yeah.
versus trying to squeeze everyone into the same off-the-rack size.
I love that analogy. And the research paper also highlighted the crucial role of TRs in development. Even slight disruptions in thyroid hormone signaling during critical periods can have long-lasting consequences on an individual's health and well-being.
So it's not just about how our bodies respond to medications as adults. It's about how our genes influence our development from the very beginning. It's a reminder of the profound and lifelong impact our genes have on our health.
Precisely. Pharmacogenomics is giving us a deeper understanding of those connections and providing us with tools to potentially address them in a more personalized and precise way.
This entire conversation has been incredibly enlightening. It seems like every layer we peel back reveals even more complexity and wonder in the world of genetics and pharmacology.
It's a truly fascinating field that's constantly evolving. The more we learn about our genes and how they interact with medications, the closer we get to unlocking the full potential of personalized medicine.
So as we wrap up this deep dive into pharmacogenomics, what's one final thought-provoking nugget you'd like to leave our listeners with?
Well, if we connect all of this back to the bigger picture, what's truly transformative about pharmacogenomics is that it's empowering individuals to take a more proactive role in their own health.
It's like giving people the tools and knowledge to become the architects of their own well-being.
Exactly.
They're not just passive recipients of healthcare, they're active participants in shaping their health outcomes.
By understanding our genes and how they influence our responses to medications, we can make more informed decisions about our healthcare and work in partnership with our providers to achieve the best possible outcomes.
It's a beautiful vision of a future where medicine is truly personalized and tailored to each individual's unique needs. This has been an incredible journey of discovery. We've explored the intricate world of receptors, delved into the complexities of gene regulation, and glimpsed the transformative power of pharmacogenomics. It's clear that this field is revolutionizing healthcare, empowering both patients and providers to make more informed decisions. Thanks for joining us on this deep dive into pharmacogenomics.